My research is in 3 different areas, all though generally around biochemical engineering. The biggest of those areas, and the one in which I've worked for the longest time, is environmental biotechnology. And in that area we try to use microorganisms and plants to solve pollution problems. The next area is the use of biosensors, actually sensors more generally, but we focused on biosensors, and these are with applications to detecting groundwater contamination and perhaps in the future to medical problems. The third area is a collaboration with some people in the Department of Environmental Health, and there we're trying to understand how toxic chemicals enter the body and move around in the body and what they do when they get to different organs, so that we can predict how cancer might be caused by certain environmental pollutants. Within the area of environmental biotechnology, one of our recent trends has been to increase the use of molecular tools, and by that I mean techniques, experimental techniques in which we can look at microbial or plant DNA and RNA and proteins, and to use those more precise, more detailed measurements to give us information and help us solve the problems that we're trying to fix. So some examples of these things in the environmental area, one really interesting project that I'm excited about is where we're using plants and microorganisms together with the idea that chemicals naturally released by certain plants can stimulate microbial activity and increase the rate at which certain soil contaminants are degraded. And in order to answer certain questions we have about that and to know which plants work best, for example, we've actually collaborated with some people in the microbiology department to look at the levels of messenger RNA that are produced by microorganisms in response to these plant chemicals. And that's told us very specifically and very clearly that these plant chemicals do indeed induce genes for pollutant biodegradation. So we're really excited about that. And another molecular tool that we've used a lot is the area of proteomics. Two-dimensional gel electrophoresis followed by mass spectrometry for protein identification. What that does is it allows us to look at a lot of proteins that are made in the cells at any given time and to understand how the cells are responding as they're degrading the pollutants. And those insights that we get from looking at thousands of proteins at a time help us understand how the microorganisms are behaving, how we can actually enhance the activities that we want. And we're doing a couple of projects in that area, and that's something I'm sure will continue in my lab for the next few years.